Welcome to Biotanemy.
Pyrosequencing.
Pyrosequencing is a method of DNA sequencing based on the principle sequencing by synthesis, in which the sequencing is performed by detecting the nucleotide incorporated by a DNA polymerase. Pyrosequencing relies on light detection based on a chain reaction when pyrophosphate is released. The sequencing process begins by labeling the DNA with biotin, which is by amplifying them with biotin-labeled primers. The labeled DNA is then attached to streptavidin-bound beads in a microtiter plate. The bound DNA is denatured with sodium hydroxide to single strands and washed to remove unbound complementary strands. Into the reaction mix for sequencing, we will add the enzyme ATP sulfurylase, adenosine 5-phosphosulfate or APS, luciferase, luciferin, and aperase. During DNA synthesis, the DNTP is attached to the 3' end of the growing DNA strand, in the presence of polymerase enzyme, leading to the release of 2 phosphates as pyrophosphate. The released pyrophosphate is used by APS in the presence of the enzyme ATP sulfurylase to produce ATP. The ATP produced is used by the luciferase enzyme to convert luciferin to oxyluciferin and light. This means we'll know as soon as the nucleotide is incorporated. And since only one nucleotide is added at once, we can understand what nucleotide is present in the particular position. The free leftover DNTPs and unused ATPs are removed with the help of the aperase enzyme.
Different types of pyrosequencing are 1. Solid-phase pyrosequencing. 2. Liquid-phase pyrosequencing. 3. 454 pyrosequencing. 4. Microfluidic pyrosequencing. In solid-phase pyrosequencing, solid substrates such as beads are used to immobilize DNA fragments for sequencing. As mentioned earlier, liquid-phase pyrosequencing will perform sequencing in a solution. Instead of attaching DNA with a glass slide or microplate, they are kept in place by attaching it to capture probes or magnetic beads. In 454 pyrosequencing, the DNA fragments are amplified within tiny droplets of an oil-water emulsion, which is attached to the beads in a picotiter plate. Microfluidic pyrosequencing combines pyrosequencing with microfluidics technology. In this method, the microbe adds with trapped single-stranded DNA within the filter chamber of a microfluidic plate, and the materials that are needed for sequencing are supplied through various channels.
Advantages of pyrosequencing. 1. Pyrosequencing provides real-time sequencing data, processes multiple samples simultaneously, and it needs fewer sample preparation steps and pre-processing steps. 2. It does not need post-sequencing electrophoresis since the results are directly converted to pyrogram peaks. Altogether reduces the time, cost, and simplifies the process. Limitations. 1. Pyrosequencing produces shorter read lengths compared to other sequencing methods, and it cannot accurately detect long homopolymers. 2. Although pyrosequencing is less expensive than some next-generation sequencing methods, the cost per base pair is higher compared to other high-throughput methods.
Applications. 1. Pyrosequencing is used in genetic mapping, genetic screening, gene regulation, epigenetic changes, metagenomics, microbiome studies, and personalized medicine. 2. Pyrosequencing is used to identify tumor-specific mutations to discover the biomarkers and thereby to develop targeted therapies.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.